Good evening, everyone. On behalf of the mentorship facilitation team, we welcome you for the monthly meeting. So we will start with the agenda. We have a um, small agenda for this meeting today. We will start with the event kickoff and the welcome address. And once that is completed, we will proceed with the introduction of mentors and mentees. And for mentors, please announce your name, batch, current location, and what is your expectation with the program. And for mentors, I'm sorry, I think I misstated. For mentees, please announce your name, batch, current location, and what is your expectation from this program. And for mentors, please announce your name, batch, current location, and the area of facilitation. And once the introduction is completed, we will move to the program highlights that will be presented by Mr. Arthur Hemachandra, and then to the monthly speaker series. Today we will have a session from Mr. Partha Anbil, who is the business area leader for healthcare and life sciences practices in IBM. Once the session is completed, we will move to the Q&A session and end this with a vote of thanks. So with that said, we will start the monthly meeting. And from the facilitation team, we thank you for joining today. So to start with the introduction, my name is Xavier Pushparaj. I'm from the 2006 Mechanical Engineering batch. I currently live in Mechanicsburg, Pennsylvania. And I'm one of the volunteers for the facilitation group. Arthur, can you please proceed with your introduction?
Oh, sure, definitely. Thank you, Xavier. My name is Arthur Hemachandra. I belong to 1969 batch, and I also did a master's degree in computer technology those days from College of Engineering, India. It was a joint program from CEG and IIT. Of course, the degree was awarded by University of Madras. And just to give you a brief introduction of this alumni mentorship program, before that, I need to clarify a few points. This is under the aegis of CEG Alumni Association of North America, which is called as CEGAANA. And we do have two mentorship programs. One is a student mentorship program, which addresses the needs of the students who are the students in the College of Engineering currently. The other one is the alumni mentorship program, which caters to the needs and aspirations of the uh, people who have passed out, the alumni who have passed out of engineering college. And we started this initiative in October of 2019, and there was a very good response in terms of registration of mentors with the diverse skill sets and mentees. And during the year 2020, we thought that we would have a good traction for this program, and we formed a facilitation team consisting of Mr. Arvindan, Mr. Xavier Pushparaj, and Ravi Goodwin, and myself. My name is Arthur Hemchandra. So it is mainly to reach out to the mentors and mentees for assistance and follow up. The current activities are monthly emails and WhatsApp text messages asking registration of new mentors and mentees are being sent. Regular follow up and feedback on the mentor and mentee interaction and process improvement. Monthly meetup of mentors and mentees with a keynote address on relevant topics from an expert to increase the visibility among the alumni. Today we have Mr. Partha Anbil. He comes with a very diverse experience and expertise in the healthcare area. And we do not do any matchmaking between the mentors and mentees. The mentors register in the website, and mentees register in the website. Mentees have a visibility to the mentors, their skill sets, and where they live, etc. And they on their own reach out to the mentors and establish contact. And if they are comfortable with the interaction, then they continue to have the mentors guide them in their areas of need. So this is what the program is all about. Any questions so far? Okay, thank you, Xavier. I hand over the podium to you.
Thank you, Arthur.
Welcome.
So we have around 16 participants. Let's take a few minutes to
Okay,
announce the
okay,
mentors and mentees.
okay, sure.
So Aravindan, do you want to go next? Hey, Aravindan.
Aravind, you may be on mute, Aravind.
Okay. Yes. Hi, this is Aravindan. I'm one of the facilitators of the mentorship program. I live here in Washington D.C., between D.C. and Baltimore area. So thank you all. I mean, this is a very timely topic, and thank you for Partha for volunteering to talk about this. I hope we'll get more mentors signed up and mentees signed up, and this is a good way to bring the CEG, the broader CEG community together. And if the mentors and mentees are people who are considering to be mentors and mentees, if we are looking for new people to sign up, please reach out to any of the 3 or 4 of us, Arthur, Xavier, Ravi, and me. There's a registration form and so on. So looking for more volunteers. So thank you. Ravi, you want to go ahead?
Yeah, my name is Ravi Godwin. I graduated in mechanical engineering in 1991 batch, actually. And I came to US in 1992, and after a stint with a company called BankTech here in Dallas, I joined IBM, and I worked on IBM for about 21 years and decided to take retirement to pursue my dreams. So right now I'm in the process of building my own little business, ideas, and also getting heavily involved in community service. And I hail from the Saurashtra community, so created an organization to help poor children to get education back in India, getting involved in mentoring Gindi the students. And so basically I just really want to just give back to the community. So that's where I am right now. So I'm one of the facilitators here. I live in Dallas, Texas. So you're welcome to reach out to me for any kind of help that you need.
Yeah, Ravi, which part of IBM you were?
I joined in the Open System Center here in Westlake, and then I moved to worldwide sales. So I was holding the portfolio of information management.
Okay.
It was not even a brand at the time, so it was just the data replicator and so-called data joiner, only those 2 little products. So we built from there this whole entire InfoSphere brand. So I was fully involved in all of these things. We used to travel all over the world. So it was a very exciting period for me in IBM.
Thank you, Ravi.
The Omni Connect that you have been working with in life sciences, okay, even that I supported for some time.
I see.
Thank you.
Thank you, Ravi. I will invite Arun to go next.
Hey, it's Arun. I'm an 84 Hindi batch graduate mechanical, and I'm here to listen to Partha. So I know Partha for a long time, so I just heard he's coming on, so I just wanted to quickly join. And I'm also the vice chair of the Alumni Association. So I look forward to hearing his speech. And I encourage all of us to link up on LinkedIn And I have sent invites to some of you, and some of you probably are too busy to link up, but I would encourage you guys to link up on LinkedIn, especially if you are Gindi alumni. Okay, thanks. Bye.
Yeah,
Thank
Arun
you.
and I were colleagues and worked together. I know Arun for a long time. We were partners in crime.
we were colleagues as well for 3 years. Yes. So
Thank you, Arun.
So
Bruce
as people are
Purushothaman.
introducing, if you are a mentor or mentee, if you could say a few words about your who, about that also, that'll be good.
yeah, I don't think anyone is linked up with me. I might be wrong. Arthur, you might correct me, but I don't think I'm linked up with anyone on the program.
Okay. So the next, Bruce Purushothaman. Can you please introduce yourself, Bruce?
Bruce, you may be on mute.
Bruce?
Okay, we will move to Padmini Sridhar. Padmini, can you please introduce yourself?
Hi, my name is Padmini Sridhar, and I'm part of the CEGAANA board. And Arthur has been sending a lot of invites. So I looked at the topic and it was interesting to me, so I decided to join.
Thank you.
But I'm from the 1984 batch, and I work in Cisco systems. Right now, I'm just involved in the finance, and I'm the treasurer for CEGAANA. And yeah, I'm not involved in the alumni mentorship at this point, but just here to listen to Partha.
Okay, thank you, Padmini. Next one is, it's going to be Raj. Raj, can you please introduce yourself?
Hey, this is you mean Rajesh Veerasamy, right?
Yeah, the display name says just Raj. So, yeah, you can go ahead, please.
My name is Rajesh Veerasamy. I actually graduated out of Industrial Engineering Department in '95. Came to the US around '97. Worked on the financial technology firm for a good period of time. I'm in the DC metro area. My area of expertise is in financial modeling, and I built systems on the particular area. And I also mentor high schoolers in robotics. So that's the area of passion for them. Anyone who's interested in the financial modeling area, that'd be great. Thanks.
எங்க காலேஜ்
Thank you.
அலுமினி மென்டர்ஷிப் ப்ரோக்ராம்ல ஆரம்பிச்சிருக்காங்க. ஒரு half an hour மாடி போயிட்டு வந்துடுறேன். Okay. அங்க ரைஸ் இருக்கு.
Somebody,
Yeah.
can you
Ramesh.
mute, please?
Yeah. Okay, we will next move to Sivaraj.
Hi guys, I am audible.
Yes.
Yes.
Yeah, I am Shivaraj, 79 civil engineering from Gindi, obviously. I'm neither a mentor or a mentee. I am observer from India. I live here in Chennai, Partha Anbil on the banks of river Adiyar. I am full and infra guy, not a digital guy, a brick and mortar brother. So I work for an insurance company, risk manager. Now I have my own risk management company, private limited company. We are into inspections, valuations, arbitrations, and things like that. I really look forward to hear from Partha. Okay, I'm happy that CEGAANA is doing a good work. From AACG side, I am also an EC member. I look forward to work with you guys, whatever extent possible. That's it from me. All the best.
Yeah, thank you, Shivaraj. The next one to introduce is Srini Rangaswamy.
I came to listen to the series today. I am batch of Computer Science 93. I work in a semiconductor company called Global Foundries, and I am the global head for the analytics team. So I manage their analytics and advanced analytics organizations.
Thank you, Srini. Sudha Vasu, can you please start with your introduction?
Sure. I am an alumni of the Electronics and Communication Group, graduated in '85. I'm here in the Bay Area, and I'm a chip designer, doing memory designs predominantly. So I am a mentor and part of the alumni mentoring team, and had mentored a couple of folks when they had questions and when somebody was looking for an internship. So one of those interns actually made it to Intel, I think. So this topic is very interesting to me, and so I wanted to join in.
Thank you, Sudha.
Sure.
Okay, next one is going to be Venkadeshan Pakirisamy. Venkadeshan, can you please begin with your introduction?
Hey, I'm Venkadeshan Pakirisamy. I can go with VP. I'm a cyber practitioner. I'm from 2006 batch IT. I have my own startup and I'm a professor at a community college in Dallas, part-time professor. I'm here to have a listen to Partha.
So where do you actually live?
Yeah, I'm in Frisco, Dallas.
Oh, I'm
Texas.
very close to you then. So we should just connect and meet.
Yeah, sure. I'll text you. I'll ping you. What's the? It's Partha? Or?
Yeah, Ravi Garvin. I'm one of the facilitators.
Oh, okay.
Yeah, I live just south of 121, around Preston and Coit.
Oh, okay. I'm in Coit and Panther Creek.
Oh, okay. Super.
Very close.
Thank you.
Thank you.
All right. Thank you, VP. Next, I will invite y'all to introduce.
Hey, this is Ramesh.
Hey, Ramesh.
Can you guys hear me?
Yes, we can hear you. We are still progressing on the introduction, Ramesh.
Oh, okay.
Yeah.
All right. Sorry,
Yeah.
I joined 10-15 minutes late. Let me do a quick intro about myself. So I'm Ramesh Krishnan. I'm 1994 mechanical batch. I did my master's in the University of Cincinnati, Industrial and Systems Engineering. And I'm currently working in a company called Synchronous. I am the head of AI/ML products and analytics and product analytics. I live in the DC area.
Thank you. Do we have anyone who is like to introduce to the group?
I believe Yarn.
Okay. So with that said, I think it is time for moving to the speaker series. So for today's speaker series, we have Mr. Partha Anbil. Mr. Partha Anbil is a business area leader and Chief Data and Analytics Officer from IBM. Mr. Partha has over 24 years of management consulting experience, primarily in healthcare and life sciences industry. He also has multidisciplinary educational background and cross-functional experience, and also an expert member of the Healthcare World Economic Forum. With that introduction, I will hand over the control to Mr. Partha to begin his session.
Thank you, Xavier. Appreciate it. Let me share my presentation. Can you all see my presentation?
It is still loading.
Not yet, Mr. Partha.
Okay.
Can I request anybody else, others, to go on mute?
How about now? No?
Yes, we can see it now.
You see one slide or two slides? Side by side. You see one slide?
We see
Huh?
2 slides.
Two
We see
slides. The next slide has, yep.
Okay, let me change the display setting so that you can see one slide.
I think the first one.
Now, can you see?
Still 2 slides.
Yeah,
Then
now
now it's fine.
Oh, okay.
Yes, it is one slide now.
Okay, let me. So I'm from batch of 79 electrical engineering. It's nice to know a couple of my batchmates joined, and thanks for joining. Currently, I'm with IBM, and before that, I'd worked for major consulting firms like PwC, Booz and Company, and I've been in life sciences industry for quite some time. Although I'm an electrical engineer, serendipitously, not by design, I got into this industry, and I've been in this industry all through my life. I've never worked in any other industry. There are a lot of changes happening in life sciences. This is one of the change. Very profound things are happening. We are at the beginning of new era. What we called before industrial revolution, the biology is going to change that in the similar fashion, and the biology is becoming engineering. I'll come to that. Why I'm saying this. You will see a lot of things are happening because of the confluence of different technologies. Biology today is where information technology was 50 years ago. If you're an engineer and wanting to get into the medicine, perhaps this is the best time to do so. And our ability to engineer biology will fundamentally transform how we diagnose, treat, and manage disease. You would have heard of CRISPR tool, gene circuitry, how things are, different technologies are evolving. If you recall, CRISPR technology got a Nobel Prize this year, 2020. So the explosion of programmable medicines in the form of genes, cells, microbes, even mobile applications and software can improve our health. And that's what this is about. It's called Biorevolution because biology never been understood this way. We have always been thinking more from traditional hypothesis-driven drug discovery. I want to keep it a little bit high level because given the target audience coming from different backgrounds, I don't want to go into a lot of technical aspect of it. If you look at hypothesis driven is basically you do by trial and error. You may have some kind of a idea where to focus on, but the drug discovery is mostly trial and error. You look at the drug and which assay would work, and then you try to do it in the lab. Then it goes through several iterations before it goes for a trial. So that's a very long drawn, tedious, time consuming, expensive process. Moore's law, all of us are familiar with Moore's law, how the performance of chip increases every year, every 2 years, and even the cost is decreasing. Whereas in the traditional drug discovery, it is the reverse of Moore's law called Jerome's law. Basically Moore inverse it. That means the cost was going up. So that is not sustainable. Drug discovery is about $1.5 billion we are spending to get to the market, one drug. That is inefficient way of doing. So all of us are very well aware of coronavirus. Do you know that the vaccine we are talking about today, Moderna, even Pfizer vaccine, you know how it was developed? Basically, there was no actual virus being used in the process. See, normally how the antibody is done is basically you inject a small amount of virus into the body, actual virus or weakened form of virus. Your body mechanism will generate antibody. Your cells will generate antibody to attack that virus, and that's how people get immunity and you are cured. That's a old traditional way. If you really know this, this is called messenger RNA. RNA is a molecule within the DNA. And what is happening in the vaccine development, COVID vaccine, is basically it was not at all developed in the lab. There was no pipette or burette in the lab with the chemical. There is no such thing. You know, the first COVID was diagnosed, somebody had in China in late December 2019. By January 20th, China gave the blueprint of the genome of this virus. It was posted online, and that's how it was developed. Basically, it was developed. Actually, nowadays we can print the sequence of the virus and program it. Not necessarily, you don't have to develop in the lab, inject people with the weak, none of those things. That is happening in the industry. So if you look at the messenger RNA, basically it is instruction given to the cell what to do. That's what this is about. So you can manipulate biology. Biology is not anymore science. It was science, but it is slowly moving towards engineering. What is engineering? Engineering means very precision based. You can actually design, you can test it, you can learn from that. It is no longer just a matter of trial and error process. That is the biggest reason why this is. If you look at the slide, where there are 4 dimensions of biorevolution. One is basically mapping and measuring intracellular components, DNA, RNA, proteins, omics. Omics means genomics, transcriptomics, proteomics, metabolic. There are many omics. That means it is related to DNA. All these things we are studying, that is revolutionizing. Why now of all the time? I'll get to it a little bit later. Why now? What is happening? Biosystem, and you have engineering cell tissues, organ, including stem cell technologies are galloping. There are a lot of advancements happening in that. Biocomputing, that may be a little bit behind. You can already see that high performance computing, quantum computing is happening. Edge network computing is happening. Next biocomputing would be on the cell-based, DNA-based computing, retrieving, storing, processing data. Maybe this is a little bit behind, but it will catch up. And there is also biomachines. You have a lot of software interfaces between connecting nervous system of living organisms to machines. So this is not something of science fiction. This is already happening. One classic example is this vaccine. And there are so many other drugs are being developed. You would have heard of cell therapy, gene therapy. Almost if you look at any pharma company pipeline, it is no. So in pharma, there are 2 types of drugs. One is called small molecule, that is primarily chemical based. And the other is called large molecule, which is basically biologics. Now, it has moved from even a drug to cell therapy and gene therapy. Literally, you take the blood from the patient, you inject certain things, basically instructions, and re-inject that blood into the stream, and that's the way it is. It is curing the disease. So, At the intracellular level, there is an increasing ability to engineer molecules and pathways for myriad uses. So why it is happening today? It is because a lot of technologies have evolved. There are what was done, CRISPR, genome mapping, several decades back happened. Now that is bearing fruit. Now, computing technology, that is bearing fruit. AI, ML, NLP, that is bearing fruit. There are a confluence of technologies are making this possible. That is what is happening today. This is going to be the major force going forward. Yes, you may think it may be an exaggeration. Yes, maybe, in the near future. But as we go along, within a matter of 10 years, it'll be a far different industry than you see. Another thing, folks, this is not really confined to healthcare industry. This is going to revolutionize agriculture, food products, energy. I'll give you one example. All of us know Impossible Burger. All of us enjoy. That's a plant-based. With this technology, you can get all the meat you need without slaughtering any animals. This is not fiction. This is happening today. You can create meat without slaughtering animals. I saw in CEG, one of the WhatsApp CEG batch, that absolutely identical milk is produced, not from the cow, okay? It is synthetic biology using it tastes, smells, in every aspect, it is a cow milk. You cannot even distinguish between two milk products because a cow milk is exact. Why? That is again this technology. I'm giving you some examples of what is going on in the industry. It may be tiptoeing at this point, but there are a lot of technologies available to make this happen. It is no longer, it's not only a technology. If you are, developing a drug for, say, diabetes, you have to develop a separate drug for COPD. Every time you have to adjust, optimize, doing all these things. So somebody said something, I don't know, I'm not focusing. Anyone not hearing me?
We are able to hear. I'm able to hear. This is around. Okay. No problem.
Okay. So this is quite different from what had happened in the past. That's why this is so important. It's not just a question of one industry. It's going to be all pervasive industry. Synthetic biology is another name for this. It's a nascent discipline. You can see DNA sequencing, technology, and it's not just a medicine alone. It's a science, engineering, all these things coming together. The much of the terminology commonly used in synthetic biology was inspired by engineering terminology. DNA code, etc. What is DNA? I want to give you some 101. Think of it, your grandma has a recipe book, okay, in a computer. Say your grandma passed away and she gave you all the best recipes. That is DNA. So what is the instruction in the recipe, how to make a dish? That's what this this is about. There are instructions, which is called messenger RNA, which is a molecule which has the instruction to tell the cell to produce certain enzymes or proteins. So that's what this is, the instruction. How do you, so if we can manipulate the instruction, if you know how the biology works, then you can manipulate the cell through messenger RNA, and therefore you can cure the disease. Certainly, that is the crux of this concept of synthetic biology. Let me ask you, Arthur, am I, because I know do I go fast? Do I go slow? Arthur?
You are doing good, Partha. No issues so far.
Okay. Thank you. So, have you heard of in the latest news, alpha folding technology? That was a big breakthrough, alpha folding. What is that basically? In the DNA helix structure, you see there is between two strands, there is a 3D dimension. That is how the protein folds itself, creates a 3D structure. Now they have decoded that. This has been decoded. If you look at the DNA, basically all these things are ultimately 0-1. Okay, computer at the lowest level it's 0-1. If you know how to manipulate that 0-1, the sequence, and you can genetically engineer disease or cure disease. Okay, I'll come to a lot of other ramifications. I'm not saying it is not without risk or some ethical aspects of it, but I'm talking about where this is going. And the cat is out of the bag. You cannot stop it. This is already happening. So if you look at why it's not moving. Okay, sorry. So why now? So we have the knowledge of the biology. Now, if you know the knowledge, then you can read, describe, and analyze, and you can understand, infer. On the left-hand side, there are multi-omics, that is different, genomics and all those things now available. We can understand it. And then single cell resolution, we can understand it. ML, machine learning, AI, we know. And therefore, we can develop capabilities using DNA synthesis, CRISPR editing technologies, and molecular reusable. Okay, so you can literally synthesize and edit the cells. How you do it, that's what the technology behind it. And you can literally program and design. The program and design, it's not a hypothesis driven, it's engineering driven. Engineering driven means design, build, test, and learn. We are all engineers. That's the way the industry is going in terms of how do you use that to modify. So if you have any questions, please ask me now, or you can ask me at the end, whichever works for you. I'm okay. Arthur, I'm okay if somebody has a question. Or you prefer I finish the presentation and then questions.
Thank you, Partha. Xavier, how would you like it?
Yeah, so we can ask the audience if anyone has questions.
Yeah.
I'll pause because I want people to. Any questions at this point?
Yes, Mr. Partha, this is Arthur. I have a question. And you said the drug discovery has all along been a trial and error process, but they do not, do not the pharmaceutical companies target a particular API and then they check that to be a prospective candidate for the cure?
Absolutely, Arthur. When I say trial and error, I don't mean completely leave it to chance. I'm saying trial and error means you may hit something 60% of the time, not very precisely. Okay. So that's what I mean by, and it's a very tedious, time-consuming, costly affair. And call me Partha, please. So you can see the first decade 2000 to 2010, all the CRISPR technology, all these things happened. 2010 to 2020, several technologies have come into play. You can see that's what I'm depicting here. It has become design, build, test, and learn. Not anymore in the lab. You don't create because once you know the genetic sequence, you can literally manipulate, provide instruction. In the vaccine, they print the sequence of the virus, and they get the chemical structure of the antibody. If you go to Moderna office in Kendall Square in Boston, you will never see a lab there. You will see a lot of printers, there are a lot of computers, there are a lot of robots actually doing this work. That it is literally, think of it, it gives the instruction to your cell to produce. It doesn't even modify it. You are not even injecting the virus into your body. It is literally telling the cell, please produce this one according to the instruction. Think of your grandma gave you a recipe and you are following your letter to that, and that instruction you are following. That instruction will have ingredients. That instruction will have how long you have to keep it in the stove and how much you need to pour, how you need to mix. Those things are already in that instruction. That's what this messenger RNA is all about. If you look at it, there are so many technologies happening. Gene addition, you can literally today, it's not future, it is today. Targeted gene cutting, you can cut. Gene editing, you can do. And it is today, it's not tomorrow. So the evolution of CRISPR itself from 1.0 to 2.0, it's one of the best. It started as a cutting tool. Now it's a platform. We are all talking about platform. Literally think of CRISPR as a software platform, information technology platform, where you can edit the gene, activate the gene. You can do all kinds of epigenome editing. All these things are possible. And it is going to only get better and better. CRISPR 2 is today. CRISPR 3 is going to happen. It's going to, because you can see that with the technology advancing, with AL, ML advancing, you are going to see more and more of gene editing. Don't think of editing as a scissors. This is a platform. Gene editing, CRISPR is turning out to be, although it started as a scissors, it is turning out to be a platform. What do you mean by platform? That means you can plug and play, meaning you have an economies of scale and scope. Every time you have to go to a lab and create a drug. Here, a platform, if you know the mechanism of action of a particular disease condition, then you can literally manipulate that gene in using computer. Literally the print, you can print and provide instruction and create a vaccine. A vaccine is one type of thing. If you're talking about some other disease, if you know how the biology works, coronavirus is the simplest, easiest one, and therefore they are able to do. If the other spectrum is oncology, it's a very complex disease. Okay, you may ask, oh, if this is so easy, why? Here is the rub here. Why? I can tell you. You have to have a very clear understanding of the biology of the disease. Then it's very easy to manipulate through messenger RNA, provide instructions to cell, and then the cell will produce. See, I'll give you a classic example. HIV virus was discovered 40 years back. There is no vaccine for that because there we have not understood the biology of the disease progression. You have to have a very clear, precise understanding of the biology. Then you can create, you don't have to go to a lab, you can create a vaccine. That is what is causing. If you think any complex disease, why it will happen as time goes by, people will see, they will understand the biology. The biology, the more complex the biology of disease, then it is difficult to do the editing. If you know, or even sequencing the virus, or even antidote to the virus, you can do that. You will be surprised. When coronavirus happened, within 48 hours, Moderna created this vaccine in terms of on paper, on the computer. 48 hours. They mapped every chemical structure possible in 48 hours. It is no different when December they approved, FDA approved. It is no different from they captured this in January 20th of 2019. When FDA approved, there is no difference in the chemical structure of the vaccine. And that's the way it was produced. So there are some issues, there are challenges. I'm not saying without challenges. So delivery is a big issue. See, if you're an Alzheimer patient, if you're trying to do, you need to know exactly which cell you want to send a messenger RNA to. You cannot send it to different cells. So those are all delivery issues are there. And you need to understand what is the immune response and how precise is molecular precision is important. And editing efficiency is also important, how quickly you can edit. Some of the birth defects can be cured by editing the gene properly. And so manufacturing of all these things are very costly now. As time goes by, the manufacturing cost to do this will be much less. You will see that is going to decrease. So there are potential solutions. Already people are working in this delivery system engineering, toolbox and protein engineering, AI, ML, data, manufacturing, infrastructure. A lot of things are happening, and it is going to revolutionize this field. Bio is going to eat. Bio is a software. It is not a medicine. It is going to eat the world, at least in the long run, if not in the immediate future. So, having said that, yes, there are ethical considerations. Editing could alter our. See, people are talking about already, unethically, designer babies. I want to have tall, blue eye and looking handsome. I always think that's in my next birth, all these wish list. But this can happen today. And, there are privacy concerns are there. Biology, once it is out, you cannot really do much about it. Everybody will try to do this. And, more importantly, unlike nuclear material, the technology need to produce synthetic biologies are relatively cheap and accessible. That's a big concern. So it is not without any risks or ethical consideration. It is fraught with risk and serious ethical consideration. That means there has to be a robust governance put in place, and otherwise it will go out of hand. You remember, like in China, somebody cloned a person or baby when it was prohibited. Doctor was, put behind the bars. So there are these kinds of things. Leakages will happen. That remains to be seen. Always there are unintended consequences when you have a technology. You don't think this is going to happen. Eventually, it will happen. So quickly for engineers, if you're an engineer, what you should do? Obviously, this is the fertile career prospects for engineers. That means you have to prepare yourself a little bit on biology. You have to write books, understand molecular biology, cell biology, etc. You need to know that, absolutely. More importantly, you need to really AI, ML, NLP, very important as applied to this. And then if you know how to use any of the CRISPR tools and technologies, which I showed you in previous slides, you would have excellent prospects into the medical profession. That's what, in fact, this is becoming engineering, not just a science. That's what my point about this presentation. I deliberately did not go into a lot of technical aspect of it because given the audience, I didn't want to bore you with that. With that, I'll take any question at this point.
Yes, Mr. Partha, can I ask a question?
Please call me Partha.
Thank you, Partha. Yes. So you were talking about the gene modification or the gene tailoring to suit to basically to create antibodies within, so that it can act against the viruses and act as a vaccine also. So does that create a problem or a side effects or goes in a different direction?
It can happen in a complex disease condition. Coronavirus is a very simple sequence of the genome.
Okay.
Whereas if you're talking about complex oncology, okay, one has to be, that's what I mentioned. It's very important to know, even within oncology, different tumor types are there. If you do not understand the biology of the disease, this technology may not work very well, or it may have some adverse consequences. You have to know the biology of diseases. See, certainly common disease like flu, it's going to be, flu will be eradicated through this mechanism. There are different strands of flu. Oncology is tough. And now a lot of research is being done. See, cardiology, there it is relatively easy. It can even produce what can I say, blood vessels. Through this process, a damaged blood vessel can be replaced. It will produce a new blood vessel. If you're talking about Alzheimer's, it's a complex thing. You may not be able to, there may be some unintended consequences. That's what I was saying. One of the things is delivery, still a long way to go. You need to understand which cell you need to target for this. If it is a complex disease, that's where the challenge is. I'm sure down the road, people will figure out.
Thank you, Partha.
Partha, this is Aravind, and I have a question.
Sure.
So if we just limit ourselves to something simple, right, like photosynthesis, I'm just thinking about, if there is, what are the implications in energy, converting solar energy to other, using biology. I don't know if there are, if we can replicate photosynthesis, artificially and, as a way to tap energy. Is there any work being done there?
Unfortunately, I'm not from energy sector, but absolutely, I don't know specifically photosynthesis, Aravind, but this has got far-reaching implications for energy sector. The first is healthcare. Healthcare and agriculture and food industry is going to revolutionize this. And then the third is energy. Fourth is, there are four different areas. Energy is certainly part of that. I forget, but I don't know exactly whether it is photosynthesis or some other fashion. Since I'm not from the industry, I don't want to profess to know that. But if you're asking me, I can tell you, I can even send you some articles which I came across in the energy sector.
Partha, this is Padmini. So my question is, what exactly is IBM's role in this biorevolution?
IBM role is, excellent question. See, this is still a nascent field for many people. IBM role is in the AI, ML, NLP, creating that kind of a platform to analyze all kinds of data so that we can create a feedback loop, how disease mechanism is, and therefore you can understand accordingly sequence the virus, and do that. So IBM role is primarily in the analytics space at this point, but many pharma companies will use IBM type of companies to help them, because if we are talking about J&J, they will create a platform and they will do that with the CRISPR technology, other allied technologies to manipulate cells and genes. And as part of that, a lot of data will come from multiple sources, not only EHR, EMR, even think of it, you are wearing Fitbit. All those data will be analyzed to, therefore, you will have a much better understanding of the disease mechanism.
Okay.
So it's more in the biocomputing area.
It is, you see, biocomputing is still behind, regardless of biocomputing, so one thing I should caveat, whatever we are doing, we are only using computer circuitry as a way to solve the problem. That has very limiting effect. So you have to think in terms of cell supremacy. What happens in the cell? How cell operates? I don't think we are there yet. IBM is not. Nobody is there. And you have to create cell circuitry way you need to think. That is possible only if you have a DNA computing. It's going to happen maybe 20 years, 30 years from now. And right now, it is not biocomputing. It is computing applied to biology.
Got you. Okay.
Partha, can I give a shot to you, Partha? Shivraj.
Sure, Shivraj, of course.
Yeah, it was a very nice unethical marriage of biology and engineering. I really enjoyed it. It was like going through a science fiction, Partha, right in the morning in India. Thanks for this experience. Aja, my question is, it looks like there is no molecules being developed. It's all done in a synthetic way, and you can always come in. Meaning, all this trial and error, what you do in a laboratory, which will happen over years, that is being cut short now by this synthetic stuff, and your time is reduced. The drug can be produced immediately, and it can be administered at the different levels. I just want to listen from you about the regulatory side that is pulling down these revolutions, and also something on the pricing side, ethical and pricing side, if it's possible to share. Go ahead, Partha. It was a nice presentation. I look forward.
Thank you, Shivraj. In fact, you will be surprised to know that gene therapy and cell therapy are precursor to mRNA. Precursor. There had been a lot of approvals given by FDA for gene therapy and cell therapy. There is a Kite pharmaceutical that is 12 billion, today market cap. Juno 9 billion. Avis 8.7 billion. Spark pharmaceutical 4. These are all approved drugs by FDA. If only it is going to expand. Yes, in terms of cost, that is a question everybody is asking that. So I don't have a real clear-cut answer for that, and that because the payers also involved here. There are many stakeholders with their own agenda here. I was more focused on the specific technology, how this is going to change the landscape. So if you look at the COVID vaccine, Moderna, it was actually developed, as I said, in 48 hours. Will you believe that? Number one. Number two, will you even believe there is no lab? No lab. You go to see if somebody from Boston, try to go, if you know somebody, go to Moderna office and see. No lab, no chemicals, no assays, none whatsoever. You will only see robots, printers, etc. And they understand the complete chemical structure of the vaccine, and then they give it to the manufacturing for the vaccine. It's all which chemical should go. Think of grandma dish and you have got the recipe and all the instructions, they create the instructions for the RNA and then they inject that. So the Moderna vaccine is basically the set of instructions given to the cells. That's how it is.
Yeah. I do understand. I only wish and hope that ethics comes in commercial pricing also, so that developing countries also get benefited by these innovations. Good, Partha. Okay, you can share this presentation to us in the any moment. Yeah.
Sure.
Thank you.
I
Thank you
have
so
one
much.
question before we go. This is Ravi. Is there a possibility of the industry inventing or coming up with a vaccine for all kind of viruses? Like
Yes.
one shot when you're born, something like that, and you don't have to worry about viruses anymore?
Yes, vaccine is one area because this is a platform and you can create multiple other vaccines for different types of disease conditions using one platform. Think of it this way. You are only changing the 0-1 sequence in the cell. Therefore, you are able to create that protein. And basically, it knows which amino acid and what kind of molecule it creates, and how do you the mRNA molecule, which cell you need to focus on. These are all the things. So vaccine is most of the ordinary regular diseases where we have good understanding. We have already done a lot of research in the area, measles and things like that. This will be used to create a vaccine. If you're talking about as we move to the complex disease conditions, that's where we need to think of it'll happen, because delivery to right cell is very important, and that's where more work are being done. It is not yet ready for complex disease conditions.
So thank you.
Partha, one question. You talked about a small molecule, which is essentially a chemical compound, and the large molecule is basically a carbon chain that is modified to get the desired effect. Then the third one is the mRNA modification, correct? Is my understanding?
Third one is more towards gene and cell therapy.
Therapy, okay.
And then fourth is now, see, gene and cell therapy is you are still doing some modification, okay? Whereas in messenger RNA, you are not doing anything. You are instructing. You are literally sending a code, software code. Think of it this way, just for a convenience. You are telling the mRNA will tell the cell what to do. You are not even manipulating, doing anything. In gene therapy and cell therapy, you are manipulating there. Here, you are not even manipulating. You are giving instructions to the cell what to do, and cell does this, and it creates an antibody based on your instruction. Think of software code that is injected into your system, and it tells what to do.
Okay. Thank you. That is it like modifying the axons and the neurons, the electrical potential, etc.
That is even more granular level. It doesn't go to that level. It goes to the cell level, molecule level. See, messenger RNA is a molecule. And cell is that, and then DNA level. It doesn't go into the electron. Doesn't go into that. Sorry, I don't know that, but to the best of my knowledge, no, not that granular.
Okay. And you talked about CRISPR technology. Stellar is what do you call it? Stellar is also a technology that can be used for the mRNA modification?
I know Stellaris. I vaguely remember. What does it do, Stellaris? I don't remember now.
Okay.
Can you give me some idea about Stellaris?
Because when I read, I understood that a Stellaris is some kind of a technology where, again, what we do is change the DNA structure to get into the desired mode or effect.
I don't know, but see,
Okay.
these are all the different forms of the platform, CRISPR
Okay.
platform.
Okay.
And
Thank
you will see more platform coming in, or more tools will come in.
Okay.
In terms of editing the gene, instructing the cell. Those things are going to happen already. That is predicated on large amounts of data it can process. That's where high performance computing is very useful.
Very good.
See, I'm not a medical, I'm not a scientist. So all my lens is through the technology and how some level of understanding I have disease, but not to the, like, I'm not a medical doctor,
Okay. Yes.
Yeah, CRISPR is the leading technology, but there is also Editas, there is Sangoma and Intellia. There are 3 others that are coming up, So if you check up online, all the stocks have gone up recently, the last couple of months. But CRISPR is the leading.
Leading.
And not only that, if you look at all the Nobel Prizes given, about at least 50 in the last 15, 20 years, many of them are indirectly or directly related to this area. Either it is data analytics or some way it is directly related to the bio related area. Indirectly, if not directly. Any other questions? I want to be conscious of the time. Xavier, I'm okay, but I don't want to.
Thank you. Thank you, Partha. We can probably limit to a couple of questions from the participants.
I will send this deck. I have more slides. I didn't want to. There are more slides I didn't show. I wanted to be very high level and tell you what. Happy to connect. Reach out to me if you have questions. My email is there, partha.anbil@gmail.com. My phone number is there. I'm also on LinkedIn. So glad to connect. Any other questions I can clarify?
Any questions from participants? But I do have one question. This is Xavier.
Sure.
So what is the participation of in terms of the industry right now, right? So what is the participation of the technology companies versus the pharmaceutical companies and the payer market?
Very good question. Right now, only the biopharma companies are looking at that. And I think eventually this will go to technology companies like Google's. See, for example, alpha folding is done by Google. Okay. They are already in it. So almost every technology company is looking at healthcare. Amazon is looking at it. They are bringing all these technologies. Alpha folding, which is a 3D decoding of the protein, structure. The connecting rod between two helix. That is the 3D structure. Now they've figured out, decodified that. That came from Google. So technology companies will play a big part in this.
Thank you, Partha.
Yeah, right now CRISPR is like partnering with companies on rare diseases, right? So money does not get spent on rare diseases because it becomes expensive where there are very few population, like thalassemia, for example. You can read online. So so CRISPR technology is used in quite a few of those areas. I think Vertex is one of the key partners, Vertex Pharmaceuticals.
Yeah, correct. Vertex is one of the key partners in the cardiovascular. Actually, they are doing a lot of work using gene therapy. Here is what I would say. The cost is very high at this point, but as any technology, the cost will come down. More and more companies will embrace and doing this. And delivery of this particular messenger RNA to the right cell is little complicated unless it's a very well understood biology of the disease, and that will eventually slowly will improve, and there it's very cost prohibitive. Those are all primarily, I would say today it's high cost, but it'll come down.
And also there have been failures. You can Google and see. The CRISPR stock, one time it fell down a bit because there were some failures, right? Because gene editing is not necessarily easy, right? So there's going to be some trial and error. So
Yeah, sure. Any new technology will have some ups and downs, and it'll have some failures. But you can see the trajectory now. At least there are some indications. The biggest thing was everybody was astonished how this vaccine was produced, and we had a print. Literally, it was printed, digitally printed. Nobody had the virus. The actual virus was never used for any of these things. Literally, it's a printing, digital printing of the sequence is what is being used to create vaccine.
Amazing.
Thank you, Partha. That was very insightful. So, is there any questions from the participants? If not, I will invite Ravi to proceed with the vote of thanks.
Thank you,
Ravi.
Thank you, Partha, for a very insightful and excellent presentation you made today. I didn't know a lot of information. The way it is going, the discovery of vaccines, etc. A little leery about taking vaccine itself. Now, I feel good about it because this kind of information was lacking for me. Thank you, and thank you so much. Also, I thank all the participants, and we are seeing a good number of new members. I hope we are able to bring in more people here and make this forum as something equivalent to IIT's alumni network in the US. Okay, we need to become very powerful, help each other, and build a good ecosystem, and become successful entrepreneurs in the days to come. So let us get to that mission and invite all your friends to join here and be together as a family. Okay, with that, I conclude today's session. We'll meet next month. Thank you so much.
Thank you for the opportunity. I appreciate it.
Thank you.
Thank you, everyone.
Thank you.
Partha, you can end the meeting.
Sure.
Thank you.